Hi everyone. So today we will be studying about a very important topic which comes frequently in all competitive exams. This will be our lecture 1. So please guys cooperate. So let's start. We have the first topic is cell cycle and cell division. What happens is there the G1, which is a gap phase, and we have synthesis phase, we have G2 phase. In gap phase, cell growth, RNA and protein synthesis all takes place. There is a restriction point which decides division by cyclin D or CDK 4,6. In the synthesis phase, there is like S phase, synthesis phase, where DNA replication, histone synthesis, and chromosome duplications all take place. This is by cyclin D, CDK2. And in G2 phase, this is basically a damage checker, which is cyclin B, CDK1, it takes place. In G0 phase, which is a resting phase, cell exits cell cycle. Mostly it happens in neuron cells or muscle cells, etc. So the next we have is G1 as checkpoint, which checks cell size and cell growth factors. Basically it checks all the growth limiting factors which takes place, like DNA damage. We have P53, P21, RB protein. In this lecture, we will study in deep about P53. The next important checkpoint is G2M checkpoint. So, what happens in G2M checkpoint? It is DNA fully replicated or is DNA damage repaired. We all find these things here. Then we have spindle checkpoint where chromosomes act like attached to spindle, which prevents aneuploidy. We will come to a detailed conclusion about CDKs in our next chapter. So for now, we will be skipping CDKs. We will come to prophase, which is a part of the part of mitosis and meiosis as well. What happens in prophase is chromatin condenses, spindle fiber form centromere, move to poles, equational division takes place. We have pro-metaphase as well, where nuclear envelope breaks, kinetochore formation takes place. We have metaphase, where chromosomes align at metaphase plate, spindle checkpoints get activated. We have anaphase, where sister chromatids work. We have telophase where nuclear envelope reforms, chromosome condenses, and cytokinase is cytoplasm divides in animal cells, which is actin myosin ring, and in plant cell we have cell plate formation. Meiosis is again a reductional division, which is M1, M2 mitosis. M1 in M1 phase of mitosis, all the reductional things happen, like homologous chromosomes get separated and equational like sister chromatids get separated. We have prophase 1 of mitosis 2 has leptotin, zygotin, picotin, diplotin, and dikinesis, which is remembered by mnemonic LZPDT. We have a feature which is mitosis is only one division, but meiosis is two, because in meiosis what happens, in mitosis we have one, but in meiosis two, because there is an sequential division. M1 and M2 takes place. There is a sequential division. Okay, so we have daughter cells, two in mitosis, but in meiosis it's four. Ploidy is diploid and it's haploid. And crossing over, no, we don't see crossing over, we don't see genetic variability, but here we see. And we find in where it occurs in somatic and here in germ. Okay, so P53, we will study in detail about P53, which is a tumor repressor protein. Tumor repressor protein, very important topic for your competitive. So the gene is basically T53 gene, which is 17p13.1 chromosome, which is a chromosome reading basically. acts as transcription factor, maintains genomic stability. It stops cell with damaged DNA from division. The steps follow is: first, DNA damage occurs, then ATM ATR kinase activates, then phosphorylates the ATM and ATR phosphorylates P53, which escapes degradation, and then it finally goes to nucleus where P53 binds to DNA, activates targeted DNA or gene basically. What is phosphorylation? Phosphorylation is stabilization, because the amount of phosphate, the amount of activation. Now we have P53, we have a terminal region which is N terminal and C terminal, and we have a DNA binding site. Cell cycle arrest. So we will study about cell cycle arrest, how the cell cycle stops. In P53 activates P21, which is CDK, N and A. Role P21 is DNA inhibitor. Then we have cyclin E, CDK2, and cyclin D, CDK 4,6. I'm reading 4,6, but it's 4/6. DNA damage. What happens first? DNA damage is spotted. DNA damage is spotted. Then P53 gets increased. Then P21 gets increased or activated, we can say. Then cyclin CDK inhibits, and then G1 is arrested. Pathways. There are many pathways, many mechanisms through which DNA gets, P53 gets activated, or maybe the cell gets arrested. So DNA repair. First, we will study about DNA repair. P53 activates gene by GADD45 or P5352. These all are not to be memorized, but to be remembered that this act and P53 activates gene. So why this happens? It's repair success, which is equal to cell cycle resume. Then we will study apoptosis, which is if it is irreplaceable, then we have number one mechanism is an intrinsic mechanism, which is a mitochondrial pathway. What happens? P53 gets activated, then Bax, Puma, and Knox gets activated, increases mitochondrial permeability. These all are functions basically. Number one function, number two function, and number three function. It increases mitochondrial permeability. Then it releases cytochrome C. It activates caspases. Caspases play a very important role in apoptosis. Then we have represses anti-apoptosis gene like BAC. Wait, let me change the color. Uh, okay, guys. Let's. Okay, it represses anti-apoptosis gene like BAC2 or BCL2 and BCL-XL. P53 shifts towards cell death, which is apoptosis, anti-apoptotic gene. P53, the mechanism we will study of P53, which is what happened. DNA damage occurs, then ATM gets phosphorylated to CDK2, then CDK2 again phosphorylates, and then phosphorylates the P53. And what the same mechanism goes to ATR. What happens? It gets phosphorylated and it's again go to the P53 and phosphorylates it. What happens when the P53 is phosphorylated? Cell cycle gets arrested, DNA gets repaired, and senescence occur. Okay, these are all the oncogenic activities. This diagram will be the most relevant diagram you will be making when you're writing any like all those examinations, college examinations. Sorry. Okay. So can you make out what happens? DNA damage by P53. This all environment. This all environment are responsible. Then there is a cell cycle. You can see the cell cycle clock here, and then we have all those metabolic pathways. What happens when cell cycle gets arrested? There is a programmed death, and what happens in the programmed death? There is like one second. There will be necrosis, there will be apoptosis, there will be ferroptosis, there will be necroptosis. So there is another term, necroptosis, which is very new to us. We will be studying about this in detail when we will study about necrosis or ferroptosis or apoptosis in detail, because apoptosis is not just p53, but it also consists of another gene mechanisms, chromosomes. Senescence, senescence, permanent cell cycle arrest. Cell alive but cannot divide, because permanent uh, because there is a permanent change, physiological, prevents tumor formation. P53 and cancer. So if P53 is mutated, then there will be cancer formation, and the cancer is we have written cancer, what are they, like all those breast, lungs, colon, etc. So I think this is it. You can please remember this diagram, very very important diagram. Thank you.